Enzymes are the bedrock of pretty much all biochemistry on the planet. Certainly life as we know it couldn't exist without them. So to understand life then, we need to understand enzymes, and this experiment shows us how pH can impact their effectiveness. Stay tuned, it's a classic. Now the concept here is a nice straightforward one. We can use amylase, a digestive enzyme found in your saliva and your small intestine, to digest starch into maltose at different pHs. And as that reaction progresses, we can periodically check the mixture to see if there's any starch left. And the quicker it disappears, the faster the reaction. Do that at different pHs and we should be able to spot a pattern. Nice and simple, let's do it. So there we go. Every 10 seconds, I take a drop of the reaction mixture and place it onto the iodine to test for starch. At first, the mixture is testing positive for starch, which means the reaction hasn't finished yet. Eventually, the iodine will stop changing color, and that means all of the starch has been digested. So the faster this happens, the faster the rate of reaction. We then do the whole thing again, using different buffers to manipulate the pH to find out what the optimum pH is for, in this case, amylase. And if we repeat this with every buffer I have, we get a graph that looks like this. Importantly, we can notice two things. First, that there is a single pH at which amylase works best, it's about 7. And secondly, that the graph is symmetrical. This is because it doesn't matter which way you change the pH, up or down, the changing proportions of hydrogen and hydroxide ions will alter the shape of the active site, meaning that fewer collisions with the substrate, starch, form that enzyme substrate complex, and the reaction will slow down. And at really extreme pH, that change of shape becomes permanent and there's no reaction at all. In other words, that enzyme has become denatured. And that's it. Nice simple experiment and a method that examiners do love to throw at you, so this is certainly one to have in your arsenal. Thanks for sticking with us. Give us a subscribe if you're into that sort of thing, and we'll see you next time.